In this video, I will be discussing the different stages of the cell cycle and a special type of cell division called mitosis. The cell cycle is a cycle that all cells actually undergo. And if you notice here, the cell cycle is divided into two main portions or events. The first, which is the longer one, is called interphase. And then in between interphases, very short in duration, is the mitotic phase or mitosis. Now, for the sake of having a beginning, let's discuss first what happens to a cell at the G1 stage of interphase. So, during the G1 stage, or the cells that undergo the G1 stage, usually are the cells that have been freshly made from a mitotic cell division. So, these are the young daughter cells after a previous mitosis. So, these cells are still small as compared to their parent cell because the cytoplasm has just been divided into two. So during the G1 phase or stage of the interphase, these small daughter cells will grow. And as they grow, they actually require nutrients to do so. And then there are certain checks in the integrity of their newly acquired genetic material from the previous mitotic phase. And if these genetic materials have some defects in them, from the previous replication of the parent cell, then they will be repaired by enzymes. Now, if these aberrations in their genetic materials are irreparable or cannot be repaired, these daughter cells will then be deemed as non-dividing cells or incapable of dividing. So they will stay as is until they die. But certain cells, even if they don't have defects in their genetic material, actually enter the G0 phase of interphase, wherein they stay non-divided. So most of these types of cells are the specialized cells, such as your neurons. So neurons are so specialized that once they are formed, it's almost impossible for them to enter another mitotic stage. So they stay as is. So they are now in the G sub-0 stage. Now, if the daughter cell from the G1 phase is successful in repairing its genetic anomaly, or if it doesn't have genetic anomaly at all, it may then proceed into replicating its genetic material. Now for preparation for it to divide later on in another mitotic phase. So it now enters the S phase. So in the S phase, this is where the DNA is replicated faithfully into a copy that's similar or that's identical to the original copy. So at this portion, you might already say that the daughter cell has twice as much DNA content as it had when it was in the G1 phase. So at the end of the S phase, when the genetic material or the DNA has been completely replicated, the cell will now undergo the G2 phase or G2 stage of interphase. Now, in this G2 stage, the cell continues to grow. Okay? So its metabolism is directed at growing, when in the S phase, its metabolism was directed to producing replicates of its DNA. Now it's back into growing. And once it has grown enough, there's enough cytoplasm and organelles, okay? It may then enter the short stage of mitotic cell division, as we see here. And of course, mitosis is divided into several smaller phases or stages. We start from prophase, then we have pro-metaphase, then metaphase, and then anaphase. So during anaphase, the division of the genetic material into two identical packages happens, and this is called karyokinesis, or the division of the nucleus. And during telophase, when the nucleus has already been divided and is going to be placed now into two new daughter cells, the cytoplasm of the parent cell will be pinched off into two new cells, and this is now what we call as cytokinesis. And this happens towards the end portion of the telophase stage in mitosis. So this is now the cell cycle. So the important portions of the cell cycle, of course, aside from mitosis, would be the checkpoints. And I've already mentioned the checkpoints a while ago in passing. There's a checkpoint in the G1 stage of interphase, whether the new daughter cell has correctly replicated DNA, so that will be checked. And then if it's not correct, it can be repaired. And if it's irreparable, then the cell is deemed to stay as is, not continue dividing, and it will die as is. And then there is another checkpoint at the G2 stage of interphase, checking if the cell is big enough or are there enough organelles for the cell to now do mitosis. So this is the breakdown of the number of hours of allotment to the certain portions of the cell cycle. So notice here the longest is the S phase, okay, 7 hours to replicate the DNA. Then comes the G1 phase, very important, especially in screening for the formation of tumor, okay, of certain cells. And then shortest, of course, is the mitotic phase, which is just allotted 1 hour in the whole cell cycle. And in the mitotic phase, the shortest one is the anaphase, followed by metaphase very shortly, very closely. And then the longest one is the prophase stage, mostly the preparation. And then the telophase stage is somewhere in the intermediate. So these are now the stages of mitosis. Now, just a clarification from my previous video, a chromosome is a chromosome whether it's condensed or not. However, when it's not condensed, it looks like threads. Messy threads, or those of a cotton candy strands. And we call those strands chromatin. Okay? So chromatin is very much present during the interphase stage of the cell. So when the cell is in G1, G0, S, or G2 stages or phases of interphase, the chromatin is present. And it can be seen as the homogeneous, darkly staining color of the nucleus, as we see in the picture here on top. And then during prophase, the chromatins are actually condensed now so that the chromosomes become more and more discrete in appearance. And we can see now as they become discrete that they actually occur now as sister chromatids. So you can see here the sister chromatids attached at the centromeres of the replicated chromosomes. And as these chromosomes condense, the centrioles also move to the opposite poles of the cell and start to divide. And once the centrioles are on the opposite poles, they will then start forming the spindle fibers, which are made of microtubules, which then attach to the kinetochores of the condensed chromosomes. So that we are now in the prometaphase stage. So prometaphase is regarded as the chromosomes traveling towards the center or the equator of the dividing cell. And we call this equator the metaphase plate. And then during metaphase, those chromosomes somehow stop the migration because they are exactly now aligned at the metaphase plate. So that's now metaphase. And then the spindle fibers continue to pull the chromosomes towards opposite poles. And the Shogoshin protein is disintegrated, thus allowing the disintegration of the cohesins by the separase. And the two sister chromatids, as we can see here now, are pulled apart. 4 of them go to the upper pole and 4 will go to the pole at the bottom in the anaphase stage. And finally, in the telophase stage for animal cells, a cleavage furrow will form in between the 2 new daughter cells until the cytoplasm will be pinched off, okay, in cytokinesis. And then the nucleus will start to reform. That's because the nucleus or the nuclear membrane somehow disintegrated during the prophase stage of mitosis. And now they have reformed back again. In plant cells, the cytokinesis is quite different because there is no cleavage furrow that's formed. Instead, a cell plate is deposited in between the 2 new daughter cells. And after telophase, these 2 new daughter cells will then enter again interphase, and their condensed chromosomes become uncondensed again, and then we have this, the condensed chromosomes are now visible as chromatins.